In 1965, a British scientist made a soap bubble that looked exactly like a living cell. He called it a cell that forgot to put anything inside. 60 years later, someone finally figured out what to put inside. What happens next will change medicine, manufacturing, and the definition of life. The question driving all of it is ancient. What is the minimum requirement for life? In 1952, Miller and Urey sparked electricity through primordial gas and made amino acids. A farmer's son from Oregon named Michael Jewett rebuilt the ribosome with a completely new operating code, a private channel inside molecular biology no cell could intercept. By 2025, global investment crossed $1.2 billion. Patent filings in ribosome engineering jumped 34% year over year. The world had noticed. Think of it as the Amazon fulfillment network compressed smaller than a human hair. RNA strands are the orders. Ribosomes are the robots. The lipid vesicle is the warehouse. A clinic in rural Nigeria. Power cuts, no cold chain, a coffee maker-sized device receives a downloaded genetic program and produces fresh insulin within hours. The $300 billion fermentation industry uses living, contamination-prone vats. Programmable vesicle reactors produce only the target molecule. Swap RNA, switch products instantly. Nobel laureate Jack Szostak showed fatty acid bubbles could absorb RNA, grow, divide, and pass contents to daughters. Primitive heredity. These engineers are running it in reverse. In 10 years, cancer-targeting vesicles in your bloodstream, detecting tumor markers, manufacturing a killing protein on site, then dissolving harmlessly. Clinical development is active. The same platform that manufactures insulin can produce any peptide toxin, no pathogen, no biosafety lab required, no surveillance trigger. IARPA began funding threat assessment in 2021. The window between the best and worst futures is open right now. This is just one discovery. We find dozens every week. Subscribe so you never miss the technology that could change everything.